Hello everyone. In this tutorial, we are going to understand the artificial cloning vector pBR322. Let's understand that what exactly is a cloning vector first. So a cloning vector is a DNA molecule in which foreign DNA can be inserted or integrated. In the modern era where genetic engineering is flourishing, there is a need for introducing new traits into the organisms. And in order to do that, cloning vectors can be used. So, cloning vectors are basically acting as a medium for the integration of the desirable DNA molecules. Now, the cloning vector is capable of replicating within the host cell to produce multiple clones of recombinant DNA. And there are several examples of cloning vectors, such as plasmids, phages, and viruses. Now, what are the characteristics of vectors which makes it successful for cloning purposes? It should be able to replicate autonomously. It must have an origin of replication. It must have selectable markers. What are selectable markers? Basically, the regions on the plasmid which can be used for the identification of the transformed cells. And in specific case of pBR322, it's ampicillin resistant gene and tetracycline resistant gene. And we are going to discuss about this in the coming slides. Now, a vector should also have unique restriction sites. Now, what is a restriction site? Restriction site basically are the sites which are acted upon by their respective restriction enzymes, and a cut is made over there. For example, in this case on the figure, PST1 is one of the restriction sites present. So the corresponding restriction enzyme, when it will come, it's going to make a cut over here. And the cut that is made is going to be used for the integration of the desired gene into it. A vector must also be small in size, it must have low molecular weight, it must be easily isolated and purified, and it must be easily isolated into the host cell. Now, moving on towards the plasmids, that what exactly is a plasmid, which is the basic foundation of constructing pBR322. Now, the definition of a plasmid is that it is an extra chromosomal, self-replicating, double-stranded circular DNA molecule. Now, this plasmid is present in certain bacterial cells. And this plasmid is extra chromosomal, which means that it is a DNA, but it is different from the bacterial DNA. It's not the chromosomal DNA, it's something else than that. So that is why it's called extra chromosomal. It's also self replicating, which means that it has its own origin of replication. It is double stranded and it is circular. Now, what is the origin of pBr322? So it is a synthetic plasmid and it was the first artificial plasmid to be constructed. And it is the most studied one. pBr322 is a commonly used cloning vector in E. coli. And what does it stand for? P in pBr322 stands for plasmid, which is giving it the identity that it is a plasmid. BR stands for Bolivar and Rodriguez, which were the scientists who constructed this plasmid. And 322 is the numerical designation given to it. So Bolivar and Rodriguez scientists, they were constructing several plasmids in their laboratory, and it was the 322nd plasmid, which was named as pBr322. And it is constructed in 1977 in the lab of Herbert Boyer at the University of California, and its size is 4361 base pairs. It's wrong here. And entire sequence is known, which means that whole of the sequence of pBr322 has been studied extensively. And the molecular weight of this specific cloning vector is 2.83 into 10 power 6 Daltons. Now, how is it constructed? As I just mentioned, that it is an artificially constructed plasmid. So it must be constructed from some other vectors, right? So there are a total of 3 different plasmids that have been used for the construction of pBr322. So these 3 plasmids include RSF2124, PSC101, and PMB1. So if you look onto the structure of pBr322, you will find that it has one origin of replication. This origin of replication has been taken from PMB1. There are 2 selectable markers, ampicillin resistant gene and the tetracycline resistant gene. So these regions were taken from two different plasmids. So the ampicillin resistant region was taken from the plasmid RSF2124, and the tetracycline resistant region was taken from PSC101. So all in all, these three different plasmids were used to construct this, and this was done in order to provide all the important characteristics to the plasmid for it to be successful as a cloning vector. There are around 40 restriction sites which are present in the genome of the pBR322, and almost 11 different restriction sites are present in the tetracycline resistant region, and 9 restriction sites are present in the ampicillin resistant region, which means that there are unique restriction sites which can be used to integrate the desirable DNA into it. And some of the known restriction sites include BAM-H1, HIND3, EcoR1, and SAL1. And the two selectable markers are ampicillin-resistant gene and the tetracycline-resistant gene. This means that these are the regions which will be used to identify or screen the transformed cells and differentiate the transformed cells from the non-transformed cells. So by using these selectable markers only, we will be able to find out that which are the plasmids which have now been integrated with the desirable gene. So how does the transformation actually take place? So in order to do that, we take the plasmid. Let's suppose that this is the pBR322 plasmid. This is the region of replication. Then there is this tetracycline resistant gene, and then there is an ampicillin resistant gene, as we just mentioned. And there are so many restriction sites as well. So we take basically a vial and we put the plasmids into it, these pBR322 plasmids, and we put the restriction enzymes, and then we also put some buffer in it. After a certain period of incubation, we do the next step, which is that we have this plasmid. Now, what is happening at this stage? So restriction enzyme is going to make cut over the specific restriction sites. Now there is an opening in the plasmid, and this opening is going to be used for the insertion of the desirable gene, and that will be done in the next step. So now this has the open end plasmids. In that, we are going to add DNA ligase, and we are going to put the gene of interest. So what is going to happen now? The gene of interest is going to go and get attached to the respective open ends of the plasmid, and the DNA ligase is going to ligate these ends. So after this period of time, we will be, after the incubation, after the step, we will be left with two options. Either there will be an integration of the desired gene, or there will not be any integration of the desired gene into the plasmid. For example, let's suppose that this is the plasmid, this is the ampicillin resistant gene here, and this is the tetracycline resistant gene. There is a possibility that the desired gene will be integrated. Let's suppose that we used the restriction site from the tetracycline resistant region. So in this case, there will be an integration of the desirable gene here, right? So this cell will be known as the recombinant DNA, or the the recombinant DNA. Or on the other hand side, there is a possibility that there will be no integration of the gene of interest, which means that both the genes, that is the ampicillin resistant gene and the tetracycline resistant gene, both are active. They have not been integrated with any of the gene of interest. So this is the non-recombinant plasmid. Let me try to revise it again. So this is the pBR322, which is having the intact tetracycline and ampicillin resistant gene. So we put this plasmid into a vial along with the restriction enzymes and buffer. So these restriction enzymes are going to make a cut in the plasmid, right? So let's suppose this is the plasmid. The restriction enzyme is going to make a cut. Now there is an open end. After a period of incubation, we are going to put DNA ligase and gene of interest into it. Now, these restriction enzymes were responsible for making a cut into a specific restriction site. Let's suppose that we added the restriction enzyme specific for a site present in the tetracycline resistant gene. Let's suppose we make a cut over here. So now there are two possibilities when we are adding the gene of interest. Either the gene of interest will be integrated here or it will not. So there are two types of plasmids that we have got at the end of this step. Now, what will happen? The next thing is that we are going to incubate these plasmids. Let's suppose that we are left with two plasmids, right? After that, the whole procedure that I just explained. One was where we had gene of interest in the tetracycline resistant gene because we used the restriction site of the specific region only. And the other possibility is that there is no integration of any kind of gene. So we have two different types of plasmids. Now we are going to incubate this with the host cell, that is the E. coli cell. After this interaction, we will be having three possibilities. First possibility is that the host cell got the plasmid without the gene of interest, right? Which means that this one. So there is a host cell and it got the plasmid that had no gene of interest. Second possibility is that the host cell got no plasmid, which means that the host cell is having, it did not got any of the two plasmids. And the third possibility is that the host cell got the recombinant DNA plasmid, which means that it got this plasmid, which has the gene of interest. So it's going to be this one. So there are three possibilities. But how will how we will be able to know that which are the cells which have got the recombinant DNA? Because this is the one we are more concerned about. We need to identify, we need to screen the cells which have got this recombinant DNA plasmid. So let's see that how the screening is done. So in order to screen, we are doing a procedure. So how do we do that? Let us suppose that we used a specific restriction site, BAM-H1, which is present in the tetracycline resistant gene. So just because we used this BAM-H1 and we did all the procedure I mentioned in the previous slide, after the integration of the gene of interest, this tetracycline resistant gene is going to become inactivated, right? So there will be this tetracycline resistant gene will not be activated anymore, which means that if we will grow this plasmid into a tetracycline containing medium, this will die, right? So because this ampicillin resistant gene and tetracycline resistant gene were providing resistance to these antibiotics. But just because we added the gene of interest into the tetracycline resistant gene, this plasmid is no more able to perform its resistance towards tetracycline now. It is only resistant to ampicillin. So how we are going to do that? So as I just mentioned that BAM-H1, as I told you, that it is present in the tetracycline resistant region. Now the recombinant molecule of the pBR322, which includes the newly inserted DNA molecule, is sensitive to tetracycline, right? After the insertion. So this is the first point, very important point. But this recombinant molecule, this plasmid, is resistant to ampicillin. This gene is intact, right? So this plasmid is sensitive to tetracycline and resistant to ampicillin now. But how we are going to use this trait to identify the plasmids which have been integrated with the recombinant plasmid, right? So what we will do is that when the recombinant cells, we are going to plate them on agar nutrient medium, right? So we are going to plate them here with a medium which will contain ampicillin. Now, because this plasmid is resistant to ampicillin, which means that if we will put nutrient media with ampicillin, these colonies, everything is going to grow on this. Either they are non-combinant, recombinant plasmids or the recombinant cells, right? So all the types of cells are going to grow on this. Either they have been like integrated with the gene of interest or not. So all types of E. coli cells which contain the non-recombinant plasmids, they are also going to grow, and the recombinant ones are also going to grow. But this again is not telling us about the transformed ones, right? So what we are going to do is we are going to replica plate this whole plate. This plate that we just got, we are going to make a replica of it. And then we are going to grow it in a nutrient media which will contain tetracycline. So when we will grow this, what do you think will happen? The, the cells that contain the plasmid with the gene of interest, which means that the inactivated tetracycline gene, they are not going to grow on it because these plasmids are sensitive to tetracycline and this nutrient media contains tetracycline. But all the other plasmids which contain both of the genes activated, or you can say which do not have the gene of interest, they are going to grow, right? And then what we are going to do, we are going to compare both of these plates to each other, and the colonies that are absent on the tetracycline containing medium but present on the ampicillin containing medium are the ones which are the recombinant cells. And these are the cells which contain the gene of interest. So that is how we screen them. Now, there are certain advantages of pBR322. So, pBR322 is widely used as a cloning vector due to its manageable size. And there are two antibiotic resistant genes which make the selection process of the recombinants easy. Now, please make sure that it is not compulsory for you to add the restriction enzymes for tetracycline resistant region only. You can use any restriction site, either it is present in the tetracycline resistant region or the ampicillin resistant region. As I mentioned, that there are 11 restriction sites in the tetracycline resistant region and 9 in the ampicillin resistant region. So you can use any of them, right? But the most commonly known one, which I mentioned in the previous slides, we mostly use those. So whatever you use, you are going to correspondingly screen them for identifying the transformed cells. And there are multiple restriction enzyme sites which makes the plasmid compatible with majority of the DNA. And it has a high copy number. This is the most important part. If the copy number is going to be low, then there is no advantage of it. So just because it has a high copy number, this is very, very favorable in the genetic engineering. But there is one disadvantage as well, that the detection of the recombinant cells having the newly inserted DNA molecule is time consuming. As I just mentioned, that you have to screen a lot of colonies and you have to make the master plate, and then you have to make replica, you have to grow the cells in the ampicillin resistant gene or resistant medium or the tetracycline resistant medium and so on. That is why it's time consuming.